From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. This episode was recorded on September 5th, 2023. Hello, Micronauts, and welcome to our Quality Quorum. Today is the 6th episode of Matters Microbial. Thanks for listening and or watching. I am grateful for each view and listen as a chance to share my own personal microbial mania. First, another denizen of my Wunderkammer. This is a shell taken from an oxygen-starved lake in Mexico covered in manganese. That's right. Some microbes breathe metals, and in doing so convert them from soluble to precipitated metal. And this is why the sticker you're seeing now, #OMG, overwhelming microbial greatness, is appropriate. And in addition, the other sticker, #WTM, what the microbe. That artwork was done by Rachel Webster, and I will put a link to manganese oxidizing bacteria in the show notes. Today's inspirational lapel pins. First is a very relevant reminder to be kinder and less judgmental to one another. And the second, of course, as you can see, is a glow-in-the-dark Matters Microbial pin. Because if it's glowing, it's more awesome. The first week of my microbiology class has been really wonderful. Using luminous microbes, my micronauts learned to streak a plate to single colonies, as you can see here. And in addition, use those same luminous microbes to create a dilution series, which looks like stars in the sky to me. We also observed, and this is something that the great Joe Handelsman taught me years ago, how Pseudomonas syringae can cause ice formation due to ice nucleation proteins. Have a look. I will put a link about this phenomenon in the show notes. And finally, and I like this, my students swabbed their reusable water bottles to look for what I'm calling water bottle buddies. You can see the fabulous sticker artwork that Ethan Kokak made. It's really fun for me to think about the adaptations that microbes make to life in a water bottle. Perhaps biofilm formation, low nutrient conditions, or oligotrophy. So the colonies you're looking at now that appear to glow under UV light, fluorescing, are most likely making siderophores that scavenge rare iron. This is a fun exercise for my students, and I do it every year, and I really enjoy it. But today, I am beyond delighted to introduce a microbial force of nature. That is to say, Dr. Irene Newton of the Biology Department of Indiana University, and Associate Chair, so definitely a very important microbiologist, BIM.
Hello.
Irene is interested in almost everything, but today will talk to us about the microbiome of honeybees and how that can impact how the honeybees live and prosper as a vital part of our lives. Welcome, Irene.
Thank you very much for having me.
Oh, it's such a delight. We've seen each other a lot here recently, haven't we?
Yes, we have. Yeah. We run in the same circles.
The same quality quorum, right?
That's right.
Well, I'm hoping that you might tell folks here, listeners/viewers, a little bit about your journey, since you have an interesting one, to microbiology.
Yeah. I went to undergraduate school at Swarthmore College, and I really didn't know what I was going to do. I think many students, enter college without knowing exactly what their path will be. But I actually was going to be a musician. That was, my declared major. And then I ended up taking an intro biology class that was taught by Rachel Mers, who's an amazing mentor and instructor. And she really inspired me to be interested in biology. And so I had done research with her every summer. during my time at Swarthmore. And I actually worked on fiddler crab behavioral ecology. So very, very different. But I also, in the interim, had a chance to take classes from Amy Vollmer, who is the microbiologist at Swarthmore. And she really inspired me to think about microbes that were host associated, that are host associated. And I developed a real passion and interest for how these bacteria have to adapt and evolve with these other organisms that they live on and within. And so that's, kind of guided my graduate school decision. And I joined the laboratory of Colleen Cavanaugh, who works in deep sea hydrothermal vent symbioses. I studied some of the first sequenced genomes of these bacteria that live inside of... Sorry, my dogs are very excited. But these bacteria that live within and the cells of these deep sea organisms, like clams and tube worms. And after that, I just, I decided that I was kind of obsessed with the idea of a microbe being, its environment, its evolutionary environment being influenced by this metazoan host that can interact with the broad ecology around it. And so I developed two model systems in my laboratory that we study. One is that of Wolbachia that lives inside of the cells of insects, and the other is that of the honeybee and its microbial community that I'll be telling you about today.
That's a wonderful journey there, and you know how I feel about Amy. So that's wonderful. And Colleen, speaking of forces of nature, there you go.
I've had some amazing female scientific mentors during my time, growing up in microbiology.
No, I think it's important. I make a joke of it, but it's not entirely a joke. We really want microbiology, rather than a big tent, to be a big petri dish.
Yeah.
And I really want everyone to feel welcome and included in that, because I certainly came from a place where none of it made any sense. And so it does my heart such good to see the mentoring that you've received and the mentoring that you do. Yeah. So and I happen to know a lot about the latter since I've sent some students to your institution. Yeah. So once again, you have to know the esteem for which I feel the esteem for which I feel toward you.
Yeah. Well, you got to pay it forward,
So I think it would be good for folks to know a little bit about your work. You sent some wonderful links that I'll put up about some of the public-facing information your institutions put out about your program, but I'd love to just turn you loose.
Oh, sure. Let's see. So I became interested in the honeybee microbiome a long time ago, like back in 2010, because honeybees are eusocial insects. They live in colonies of tens of thousands of worker bees. These are effectively sterile female insects. There's only one reproductively capable member in the colony, the queen. So she is in charge of laying all the eggs and maintaining the numbers of that colony. But the workers do, as their name implies, all the work. And they clean the colony, they tend to the little baby bees and also the queen, and they obviously forage. So most of the listeners of the podcast have probably interacted with foraging worker bees if they see them on flowers. But everybody knows that honeybees make honey because it's in their name, obviously, this delicious sweet thing we like to eat. But they also make this other food called bee bread. So they harvest pollen from the flowers they visit, and then they add gut regurgitants to it. And this is thought to be both, a way that they make the food more nutritious through microbial activity, but also that it keeps it preserved over the winter so that they can actually survive as a colony through the dearth of winter. So I became really interested in the honeybee microbiome because of bee bread, because of this food that, they take this recalcitrant carbohydrate that's, pollen, the sporopollenin wall of the pollen is kind of notoriously difficult to break down. And then they make food out of it. So I was really interested in exploring what microbes might be there. And that kind of got me into thinking about the microbial community associated with bees at large. And I think one of the super fascinating things about bees as eusocial insects is that you can study, how microbes are associated with an individual bee for sure. But I think what's fascinating about it is the colony context, the fact that they interact with each other socially. And we know so much about bee behavior and what these social interactions mean. The fact that they leave the colony and sample from the environment. So you can think about microbial exchange at that level. And also the fact that there is this developmental process of the bee happening underneath all of that. So they're holometabolous insects. So they go from an egg to a larva, and they have many molts as a larva until they turn into a pupa. So you can think of it like a chrysalis for a butterfly. And then they emerge as adults, and they have to be re-inoculated with the microbial community because they shed their gut lining through metamorphosis. So the microbes that they have as larvae are actually different from what they have as adults. And you have this built environment on top of it all too. So not only do you have social interactions and beautiful behavior, also this developmental process, but then the bees make their nest. So they make these wax cells, right, that they store all their nutrition and all of their brood in. And these environments are also microbial habitats, so potential microbial environments as the honeybee-associated microbes move between members of the colony and exchange within the environment of the colony and the bee itself. So my group is really interested in what these microbes are, how they might help the bee, what roles they might play in these different environments, and how these processes of both like spatio, looking at all these different habitats in the colony, but also temporal as the season change. How does this alter the role of these microbial members and also their composition?
May I ask a couple of questions?
Sure.
So I don't mean to go all Seth Bordenstein on you, but it's interesting to think about, and I don't have an elegant way of putting it. We encounter microbes in the environment, and then we carry them with us. And I don't know in my own studies where I'm looking at lizards, the microbes that we find in their gut, how many came for the prey insects that they ate.
Sure.
In a similar fashion, there has to be a core microbiome that's currently among the members of the hive.
Mm-hmm.
And they must do something to, I hate the militant expression, but to exclude other microbes that might be coming in. Do you have thoughts on that?
So it's an interesting question. I think it's true for all host-associated microbial communities. There's a question of how much contribution do environmental microbes make to the assembly of that community, and how do the existing core members is there a priority effect, for example, in their establishment of their association? No one has done this specific experiment that might answer your question precisely, which is take a newly emerged bee and then see if it can be colonized by something random, right? I think there's some elegant work that was done by Rob Knight's group with notobiotic mice that basically added, really random microbes from all sorts of places, like including, Sipowiscent marsh sediment, right? And then those bacteria could actually colonize if there was no existing community there to begin with. Which is fascinating and suggests that there is some exclusion, some way that, the prioritization of inoculation from the generations or from mother to offspring, establishes the community that's there. But what I can say is that even though the bee goes out and samples all sorts of different microbes from the environment, and it must be bringing lots into the colony, we find a consistent, conserved core microbiome in the worker digestive tract and very specific microbes associated with the queen and with developing larvae. And whether this is because of the diet and how it selects for a community, or because of the priority effects due to inoculation, the social inoculation within the colony, I couldn't answer that specific question. I think that would be an interesting thing to explore. But I can tell you that in our group, at least one microbial member, which we've been working on for some time now, called Bombella apis, it does exclude filamentous fungi from the colony and protects bees from fungal infection.
It's just amazing. We are just a community, is when it comes down to it. And so you're looking at the community of a community insect, which is really interesting.
Yeah. we're all surrounded by a cloud of our microbes, right? And so when I teach microbiology and students are sitting next to each other, cheek by jowl in these lecture halls, I try to tell them, like, look at the person next to you. You guys are effectively rubbing microbial clouds together because you're constantly shedding, right, all your microbes associated with you, and that's part of the social exchange of microbes, right?
It really is. It really is. And it's something, I watch students when they start to realize this, and this is one of the things that I fight about, is this negativity toward the microbial world, what I call the devil microbe problem. And so it's just natural. We're doing this with our water bottles, for example. The students are all freaked out by that, but they're part of their own community.
Yeah. And they're also maybe coming from the water supply itself, right? It's just,
exactly.
We're all covered in microbes. I think it's an important thing for people to understand, because so much of the conversation in the public sphere, if people know about bacteria, right, they know that they cause disease. This is how they learn about them, right? They learn it through the lens of medicine. And that's not, it's an important lens, but certainly it's a very vanishingly small fraction of bacteria that are pathogens of humans.
I agree completely. There's a wonderful cartoon that my friend Maggie Middleton put together one time describing pathogens, and I know I'm not supposed to use that word. Professor Casadevall will be unhappy with me using that expression because it's all contextual. I get that. But really, pathogens are like the juvenile delinquents of the microbial world. They're very low in number, but they get all the press. And that's one of the reasons that I'm excited to have you here today, is to talk a little bit about what you've found going on there with the microbes and your honeybees.
Yeah. So largely, I would say that my lab studies symbiosis, and I define symbiosis to be very broad and inclusive of those that cause fitness benefits to their hosts, so mutualisms, and then on the other end of the spectrum, those that are a detriment to their hosts, so what Arturo might call pathogens. And the reason it's important to be inclusive is exactly that, what you mentioned, that it's contextual, that even a microbe who you think provides a benefit in one context can be providing a detriment in another. And the other reason it's important to think of this as a larger umbrella of host-associated microbes is that the mechanisms microbes use to interact with their hosts are the same, regardless of fitness outcomes. So the molecular tools that bacteria have developed to be able to associate with hosts and to modify them to facilitate this host association are really conserved across, doesn't depend on the fitness effect on the host. So across this spectrum, it's the same.
Well, I just enjoy this system so much. So take us into your lab. Tell us what you've been finding.
Yeah. So we got really interested in Bombella apis when we discovered that honeybee queens, which are that sole reproductive member in the colony, have a very distinct microbial community compared to worker bees. So back in 2015, most of the work that had been done on honeybee microbiome had focused on honeybee workers, and that makes a lot of sense. They're the most numerous member of a colony, right? And they're also easy to sample because there's many of them. If you sample a queen from a colony, you effectively kill the colony. So it's harder to get, a lot of queens if you're not breeding them yourself. And so we were really curious to explore what was the queen microbiome. The other thing I think it's important for your listeners to know is that there's this amazing caste distinction between workers and queens in a honeybee colony that is entirely based on the nutritive environment that is sensed by a larva as it develops. So honeybee queens and honeybee workers are genetically identical, but queens can live years and workers live a matter of months. Queens are fully reproductively capable. Workers have these, shriveled ovaries and they can't really lay many eggs. It is also the case that the queen dominates the whole colony with her pheromones. The workers are kind of subjugated into doing the tasks in the colony. These are major physiological distinctions. The queen is almost a completely different organism, but any larva can become a queen if they're fed the right nutrition. And that nutrition comes in the form of royal jelly. And this is a weird substance that is a glandular secretion made by bees and fed to very young larvae, or if the larvae are going to be a queen, they get it throughout development. And then it's also fed to the queen herself. So we sampled queen microbiomes throughout development, as well as in adult queens. And we discovered that queens have this microbial community that's really dominated by an acetic acid microbe called Bombella apis. And that's fascinating and very different from workers. Workers really don't have much Bombella apis in their digestive tract. And we know now that it's really Bombella apis, if it colonizes workers, it's going to colonize in what's called the social stomach or the crop. This is the little inflatable balloon that the bees use when they go and sample flowers. I always say, like, bees aren't carrying buckets with them, right? They're going and like putting the nectar in the bucket. They are sucking up that nectar into their crops, and then they regurgitate it into the mouths of their sisters when they get back to the colony. So they say there's a lot of vomiting in a colony, a lot of very close, intimate social exchange of microbes. And anyway, so Bombella apis lives in the crop. We now know it lives in association with larvae. We know it is in the queen digestive tract. It's found in nectar also because of that crop association. And why is it found in association with queens? Because it is the only microbe that we have found in the honeybee association that can survive royal jelly. So royal jelly itself is antimicrobial. It is highly nutritious, but it has all these antimicrobial peptides imbued in it that the bees themselves add. And it's also got very low pH. It's got, this kind of jelly structure to it that is itself highly osmotically stressful for microbes. And so Bombella apis is the only organism that can replicate in royal jelly and survive this. So it's a very strong selection for this, particular microbe to be there. So my students that just recently graduated, Audrey and Delaney, they explored two different mutualistic traits of Bombella apis. Delaney discovered that, it was kind of a surprise to us that there are these few fungi in bee association, right? That we don't have very many fungi at all, even though they sample plants and pollen. And, we know that those environments are coated with yeast and filamentous fungi. We really don't find very many in honeybee association, either in the hive environment or in the digestive tract. So she wondered if there could be a microbe that was providing an antifungal role. And it turns out Bombella apis is. So it produces a really potent antifungal, and it's got several biosynthetic gene clusters in its genome that suggest it may be either a terpene or maybe a polyketide. We're trying to figure out the exact molecule and the genetic underpinnings of that now. And then the other student in the lab, Audrey, took a different path and discovered that Bombella apis produces copious quantities of amino acid lysine that supplements the nutrition of developing larvae. So you can actually drop the nutrition of larvae in the laboratory. We can in vitro rear them, and so we can reduce the nutrition we give them. If we supplement with Bombella apis, they grow to the same size as they would on full nutrition. So both of those traits make Bombella apis quite an important mutualist in honeybee association.
That's definitely a keystone role.
Yeah, it seems like an important thing for this microbe, for the bee. Yeah, and they're and really well adapted to this host association because it can grow in royal jelly. So one of the threads in the lab that we're finishing up now is trying to figure out what allows Bombella apis to grow in royal jelly. What attributes of this microbe allow it to survive all this osmotic stress.
That's amazing. I'm guessing they're overexpressing GROW-EL, but that's me.
We'll see. We have an experimental evolution study that we've done to try and figure out what might be responsible, and we've done some resequencing, and we're characterizing those strains now. So, yeah,
I have so many thoughts about the measurements of osmolarity in a different context than your stuff that I would love to chat with you about in your copious free time.
Sure.
That sounds like a wonderful project, and I'm so glad to see the students doing wonderful things with it, and you shining by their reflected light, right?
Yeah. Well, they were amazing students. They've gone on now to postdocs in other symbiosis labs, and so it's nice they're staying within the symbiosis community, and we'll probably see each other at meetings.
That's so wonderful. Do you find that the work that you do has any relationship to colony collapse syndrome?
Oh, yeah. So I get this question a lot because colony collapse disorder was very prominent in the media when it first happened, right? This is about the early 2000s is when people started noticing colony collapse. And the specific phenotype is that the workers leave the colony, they go out and forage, and they never return. And so you end up with these waves of workers that just keep leaving, and the colony gets smaller and smaller and smaller. And so at the end, when you go look, it's not like you find a big pile of dead bees and you can see if they were intoxicated or there was some kind of pesticide poisoning. What you find is that, the queen is left with a bunch of brood that she can't raise because there aren't enough workers there to actually do the job. And and in the end, what caused colony collapse? There's some hypotheses out there that I think are quite compelling, including one that it says specific pesticides that disorient bees and make it difficult for them to find their way home. That's possible. There's work in laboratories that is quite compelling based on that hypothesis. But bees have a large number of different, a panoply of stressors that make it difficult for them to survive over the winter. There are lots of chemical stressors like these pesticides and herbicides that they interact with. There's also the lack of diverse floral resources in the environment. If you think back to the early 20th century, most Americans were farming. And so they're at small family farms that were really diverse. And now what we tend to have are many, many, many hundreds of acres of monoculture crops that are wind pollinated and less nutritious for bees. And then the other thing I would say is that there are introduced pathogens and parasites that can really affect the health of the bee, like the Virology, which is this fascinating parasite that comes and sucks on the fat body and hemolymph of developing bees and spreads viruses around the colony as well. So there's lots of different reasons why it's kind of hard for bees. But I think maybe now is a good time to mention that bees are not native to North America, that they are a domesticated insect. And so when we say that bees are in decline, it's a little bit like saying chickens are in decline. Chickens are important to us, and not important to other birds, are they? And so along the same lines, bees are important to us because of how we farm, right? And we use the honey as a resource as well. But native pollinators, could do the job if we farm differently. And native pollinators don't care so much that bees are in decline, I would say.
I find that, you hit on an important point that I want to emphasize to everyone, that there's something, and again, I'm not being critical of journalism at all, but I am saying that the media, the old-fashioned expression, if it bleeds, it leads. We're all familiar with that. Yeah. But there's something about headlines that just seem to worry people. And I'm not saying there aren't things to be worried about. Of course there are. But one of the goals that I have is to be able to give more nuance and context to what we hear, especially with the microbial world. So while many people have certainly heard about colony collapse syndrome, or maybe they're allergic to insect stings like I am, whatever it might be, what they've learned from you is not simply the intricate nature of the bee community, but some really interesting facts that certainly I didn't know, having to do with the antimicrobial effects of royal jelly and things that you're doing in the lab with them in a way that I think really connects with the average person, because most of us like to put honey on our toast. And we see them kind of bumbling around the garden, and now we have a whole new appreciation for them and the work that you've done, I think.
Thanks. Yeah, I also think even though honeybees are not native to this continent, they're a poster child for insect health and for caring about the environment. And if people plant more pollinator gardens that help honeybees, they'll help other insects too. So I think they're a little bit of a canary in the coal mine in that, their declines have been mirrored by the decline of insects broadly, and people care about this furry charismatic insect. So they serve like a very positive role in that sense. In the public sphere.
Well, I really have appreciated this, and I've learned a lot, and I know that I'm going to put some links to things, and maybe I'll ask you to provide a couple more links. You already gave me some, and I'll put them in the show notes for people to learn more. I will, of course, give a link to your laboratory. I like your website very much. I'm really quite jealous. But I think as we start to wind down a little bit, I wanted to ask you. Normally, I ask 3 questions of people, and I find that they normally answer most of those questions during our conversation. But there is one thing that I hope you can share with all of us. There was one instant in your life that sold you as being a microbiologist, and I wonder if you might share that with us.
Gosh, that sold me on being a microbiologist. I think that when I determined as an undergraduate, I was trying to decide, I knew I wanted to go to graduate school. I knew I was inspired to continue in science. I was trying to decide what my interests were. And my mentor, Rachel Mers, had a great suggestion, which was to go through the body of work I developed as an undergraduate and see if there were any unifying themes that popped out at me. And when I did, I realized that in every class where I'd had some flexibility to do my own independent project or research paper, I'd always chosen a microbial symbiosis. So I did work on Riftia pachyptila, I did work on Euprymna scalopes, I did work on Rhizobia. You kind of like hit all of the high notes of like the major microbial symbiosis models that were out there. And it occurred to me that, that's probably what was most exciting to me. It was sitting right in front of my face, and I just needed to go through and figure that out. Microbes have just an amazing metabolic breadth and, capability that allow eukaryotes to take advantage of this genetic diversity and leverage that to live in environments and do things they otherwise wouldn't be capable of doing. And that's what's really always fascinated me, the abilities that microbes provide to their host, and also the fact that the microbes have to deal with this eukaryotic association, right? And navigate that relationship, figure out how to be transmitted, how to, colonize and maintain that niche. All those things have always fascinated me. So I would say that like when I was applying to graduate school was when I figured out that I'm really a microbiologist, or that was my passion.
You can't see them, but microbial tears of joy are coursing down my face. And you should know that the first tattoo I ever got says, "Ovite parvile domini," all hail the small masters, because they really do run the show. There's that wonderful Beyonce song, "Who Run the World?"
Yes.
Well, we know who run the world, right?
Right. That's right. I mean, we evolved in an environment teeming with bacteria. That's who were there first. So
that's right. First evolved, last extinct.
That's right.
Do you have any further things you'd like to discuss or bring up?
Oh, geez. Put me on the spot for that one.
I'm good at that.
I think that it's I always end with a plug for basic research, and to try to remind everybody that, sometimes we can't tell what we'll discover, right? When we turn over a rock, maybe it's, you might describe it as stamp collecting or discovery-based science, but discovery-based science has led to amazing developments that have basically been the foundation for whole fields, like the discovery of Taq polymerase, which led to all of molecular biology. So I think we should continue to turn over those rocks and figure out who those microbes are and what they're doing.
I find that this is one of the things that I wish I had a magical microbiological wand to wave, that most of the really cool things that everyone takes for granted now were kind of found by accident.
Exactly.
Accident's a strong word, but they just looked.
Exactly. I mean, people did not expect the result, I guess. And that's the point of science, right? We don't know what we're going to find.
Right. Well, Irene, it's been wonderful for you to spend some time with us today and with me. It's always a pleasure to chat with you.
Thank you very much for the opportunity.
Oh, I'm very grateful, and I will be sending you a thank you.
All right. I look forward to it.
You're awesome. My best wishes to your family and your laboratory.
Thank you so much. Yeah. Bye-bye, y'all.
Bye. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Irene Newton can be found in the biology department at Indiana University. Many thanks to Julia Russo for the superb editing and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our quality quorum today. See you next week on Matters Microbial.